guys.
Hi guys.
Hi guys.
Hi guys.
Hi guys.
Hi guys.
Okay, stop. Okay, so today we're going to learn about cells, and our teachers are Fiona, Heidi, Sheng, and Sanjeev. Yeah! We are starting off with the basic properties of cells. First off, they are complex and organized. Cells are consistent in their appearance and their macromolecular composition. Most cells have organelles like the nucleus and ER, and are usually made of similar components like proteins, lipids, and water, etc. They are organized as they have a special function such as these. Secondly, cells possess a genetic program that is packaged into their chromosomes. They are built according to the genetic info that is encoded by the genes. Thirdly, cells also can self-replicate. They usually do so by mitosis and meiosis. Fourthly, cells acquire and utilize energy. They require energy for constant development and to maintain their complexities. Cells also respond to stimulus. They have receptors on their cell membrane that interact with specific substances, and the cell will react to them, like changing its metabolism, for example. Lastly, cells can self-regulate, as their cellular activities need to have precise coordination. Lastly, cells can evolve. That is why human intestinal cells and bacterial cells look very different. Who knows what they will look like next time? Now, let's take a look at the difference between prokaryotes and eukaryotes.
Firstly, let's talk about the nucleus. The nucleus is the brain of the cell, which contains all the genetic information. It appears as a viscous mass of material, and there can be multiple nuclei in one nucleus. In a nucleus, there are heterochromatin and euchromatin. Firstly, the heterochromatin is a tightly packed form of DNA. It stains darkly with the Giemsa stain, and it is an inactive gene expression. So, the more heterochromatin there is in a cell, the more inactive the cell is. Moving on, the euchromatin. It is a loosely packed form of DNA, and it stains faintly with the Giemsa stain. It is an active gene expression. So, the more euchromatin there is in a cell, the more cellular activities it has.
These are the contents of the nucleus and the functions of its envelope. The mitochondria's outer membrane is high in lipids, and it has porous channels that are portals to the key molecules in metabolism. On the other hand, its inner membrane is higher in proteins than lipids. The proteins are important in calcium-ion regulation. The cristae are the internal membranes, and they are sites of aerobic respiration and ATP formation. Now, the mitochondrial matrix. It has ribosomes as well as mitochondrial DNA. This mitochondrial DNA is different and more specific than nuclear DNA, and it codes for proteins that are only used in metabolic processes. The rough ER. It has a rough appearance and is basically a network of flattened sacs called cisternae. The first function of the rough ER is to produce protein. It has molecular chaperones to ensure proper protein folding, and has the necessary protein processing enzymes like protein lysophile-isomerase. Now, moving on to its second function. It is to secrete proteins. Hence, in protein-secreting cells like the pancreatic and salivary gland cells, more rough ER is present.
The smooth ER, namely the smooth endoplasmic reticulum, has no ribosomes and it has tubular membranous elements. It has various functions. Firstly, it is involved in the synthesis of hormones such as steroids in the endocrine cells of adrenal cortex. Secondly, it is involved in the detoxification in the liver, such as for ethanol. And lastly, it helps to regulate the release of calcium in skeletal and muscle cells. and as such it is extensive in cells such as muscle cells, kidney tubules, and
Next, let's talk about the Golgi complex, also known as the Golgi apparatus. It is a flattened disc-like membranous cisternae arranged in orderly stack, and it is involved in post-translational modification, which involves assembly of carbohydrates components of glycoproteins and glycolipids. It is also responsible for transport of proteins. The proteins are out of the cell via cell surface. This is also known as the vesicular transport model.
Lysosomes are the cell's digestive organelle. It is variable in size, contains diverse variety of enzymes, has low pH due to acid hydrolases, and its function is to break down material brought in from external environments such as bacteria, and also for organelle turnover.
Peroxisomes. They are also known as microbodies, and their main function is to degrade hydrogen peroxide, which is a toxic region produced by enzymes which need oxygen to oxidize substrates.
Endosomes are formed when they bud off from cell membrane or from the fusion of numerous vesicles. Endosomes are also involved in protein trafficking via linkages with the Golgi complex, and it is also involved with the recycling of cellular waste that is produced by the degradation by lysosomes.
The functions of the cytoskeleton include dynamic scaffold, which serves as a structural support, internal framework, which allows for spatial organization and positioning and attachment of organelles within the cell, network of tracks, which allows for direct movement of materials and organelles within the cell, locomotion, which brings about force generating apparatus and allows cells to move or migrate, and lastly cellular division, which allow partitioning of organelles and materials.
And thus, this is what we have to memorize.